So, last speaker in this section will be Pascale Gaudet from Swiss Institute of Bioinformatics, and she will present nextProt protein database.
Good afternoon, everybody, and thank you for the organizers for inviting me here. It's a pleasure to be here. So just to start, because there's not enough time, I'm curious to know how many people have used the biological databases in their work. For example, Swiss-Prot or Ensemble, or is it most of you? Some of you. Okay. So, you know, with like all the new high throughput techniques and all the, the sequencing of the human genome and all the different species, and now they're sequencing individuals. So there's this project in the US, 1,000 genomes. There's a lot more high throughput data everywhere, and to make sense of this, there's a lot of biological databases out there that exist to organize it and make it publicly available. And what I'm going to describe today is nextProt, is the database that I'm working at with Jonas, so I'm responsible for the scientific content of the database. And just to give you a little bit of perspective, because I think it's very interesting to see us, also all the people here, and how the industry and the academic world is interacting. So just to explain how we're set up. So nextProt is one of the resources of the Swiss Institute of Bioinformatics. And the Swiss Institute of Bioinformatics was founded in 1998. The main resource there was Swiss-Prot. It's quite well known. It describes proteins of over 200,000 different species. There's millions of sequences in there. And there's also about 20 other groups that have all kinds of resources, for example, for protein interactions and for networks and so on. And so the nextProt, specifically resources, was founded by Amos Barak, who's also the founder of Swiss-Prot and of the Swiss Institute of Bioinformatics, as a resource targeted really towards human proteins. So Swiss-Prot is very nice, but the problem is, it's very hard to model all the different data we want to put in the database and trying to be representative of all the species. And so what he did is he took the human chunk of Swiss-Prot and developed it into a new database. And then we're expanding from that to include new data that is of interest to the pharmas and all the medical applications that are more relevant for human biology. So this is the front page of nextProt. So really what we have is we have a publicly available website, and the information we put in is all publicly available and free. So like I said, it's a central resource for all human protein-centric data in the component Swiss-Prot. So this was mentioned before, but if you go from the genome to the proteome, taking into account the alternative splicing, the different post-translational modifications, and also between different individuals, it's becoming very clear that there's a lot of variance at the gene level, which means a lot of variance at the protein level. So the number of different proteins that exist out there is like 1.5 million was mentioned before. We have a calculation of 5 million, but if you take also into account the combination of, maybe when you have one mutation and, one variation and a different variation, maybe that disrupts certain interactions and so on. So the combination of all the different proteomes of all the different human beings that exist right now is humongous. And so, so this is the status of the data that we have right now. So it's 20,000 entries containing 16,000 isoforms and over 300,000 variants, and so and 80,000 post-translational modifications. So we get this data from either collaborators or from other public resources. So for example, the variants we obtained from DBSNP, which is a database in the US from the NCBI, and also from COSMIC, which is a database in the UK funded by the Wellcome Trust Center. And so, essentially what we're trying to achieve here is to have a one-stop shop for all the human proteins, okay, of Swiss quality. And I'm Canadian, but I can see, like the Swiss quality, the Swiss are known for watchmaking and so on, the precision. And so as you probably are aware of when you read the scientific literature, there's a lot of data, and we don't want all the data. We really want to have the cream on the top that is really high confidence, and you can take it and go away and be confident that what your next hypothesis based on that will be, you will trust it. So, and then the other thing that we haven't developed very deeply yet is to allow, complex querying and, different tools to understand all your data. And so, ultimately, we want to help the community to do their work by having this central resource. So, I'm just going to go a bit quickly on some of these things, but just to talk about the high quality, this is very central for us, and what we have done is this Olympic, again, system where the data is graded as silver, gold, or bronze. And so again, so the gold data is really the really high confidence data, so usually around 99% confidence, especially in proteomics set where there's actual quantitative value attached to each data point, we can really make a clear cutoff. The silver data, because it's interesting to have some other data, to allow you to explore. So we do take data in which we're a bit less confident, but we never go below like 90% confidence. And then bronze data is what we don't integrate. So there's only 2 winners here. And so what we do is for each data set or each resource, we evaluate all the data either directly, or if we can collaborate with the data providers, we ask them, how they feel about the data, like because they also know, like if you run a big mass spec experiment, what is the best set and what is the lower confidence set and what they wouldn't trust but they keep in because they think, after further validation it may be interesting. So we do collaborate a lot with data providers. And so the user interface is divided into several views. So you can see on the left-hand side here, you can view, so I'm just going to go a bit quickly, but it's divided into different perspective of the protein, if you will. So the function, just generally describing the function, the medical view in this case, well, in this case it's insulin, and some variants are associated with diabetes, but we don't have a disease diabetes because it's not the insulin that's causing the diabetes, it's the receptors or the expression. We also have a view, and this is data that we have specifically in nextProt, not available in Swiss-Prot. It's from the Human Protein Atlas, developed in Sweden by Matthias Sweden, where they've done polyclonal antibodies against about, right now, 6,000 human proteins. Their goal is to do the whole human proteome. And they've done staining on all normal human tissues. They've also done staining on cell lines and on diseased tissues. So we have all the data from that resource, so you can look at your favorite protein and see more or less its expression profile. Yeah, so what I was meant to show also is semi-quantitative, so it shows you if there's high level or low level of expression. We also have a view for the sequence, which is linking a graphical diagram of the protein. So here you would have, for example, domains. This is a transmembrane protein, so it's got cytoplasmic, transmembrane, cytoplasmic, membrane, cytoplasmic, and so on domains. Metal binding sites here. I don't know if you can read this. The active site is there, and then any variant in the sequences. And in this case, there are no post-translational modifications. But like Jonas just mentioned, if there were phosphorylations, those would also be shown in the graph. And then when you click on any of the section in the graph, it highlights in the text box here what you're looking at. Also, if there's a publication, it will show you the reference for that information. And it highlights in the sequence exactly the section you're looking at. So if you click on one phosphorylation site, it will show the paper or the papers in which the site was demonstrated, and then the exact position in the sequence. Yeah, so that's a thing I'm going to use my 1 minute for. Is we have a collaboration with the Human Proteome Project, HUPO, And so what they aim to do is to understand the whole human proteome. So that's a big multi-natural collaboration. And they have like what they call the 3 pillars. The mass spec community is providing a lot of data. They're trying to identify all the proteins in human. The antibodies, like I mentioned, that's mostly the work done by Matthias Hülen in Sweden. And then the knowledge base part, and this is nextProt. So nextProt, it acts as the knowledge base axis of the human protein, the Human Proteome Project of UPO. So that's a lot of abbreviations. But so the goal is for all the groups to eventually forward all their data to us so they can compare all the information that we generate. And so while we have generated special views for people doing proteomics that give extra the information about how the experiments were done, which type of mass spec was used, which tissues were used, and so on. And we also support what is called the PEFF format. So when you're doing proteomics, it's a kind of FASTA file. This is very technical, but this is a FASTA file where you have information about variants and post-translation modifications within the file. So when you're doing proteomics to do the analysis, it's nice to have all the information in a single location. Yes, I'm finished. I'm just gonna say that's one out. Also, this is just just the team that works with us on this project. Thank you.
One question.
So what is the main criteria to give gold or silver medal?
Yeah, it just depends on the rate of the estimation of false positives. So if we think that there's about 1% false positives in the set, then it'll be gold.
Thank you very much. And so we conclude this section, and I give the seat to the next moderator.